Let's talk immunology. Have you ever wondered how your body can just absolutely crush a common cold in a few days, but then a tiny little paper cut can sometimes get dangerously infected? Well, it all comes down to your immune system, this brilliant, super complex army of cells and signals. Now, for the USMLE, look, mastering immunology isn't just about memorizing a bunch of facts. It's about really understanding the story. And today, that's exactly what we're going to do. We're going to follow that story from the very first sign of an invader all the way to its defeat. And we'll hit every high-yield concept you absolutely need to know. Let's get into it.
So here's our game plan. We're going to start on the front lines, looking at the innate system versus the adaptive system. Then we'll dive into the MHC system. Think of these as the cellular billboards our body uses to signal for help. After that, we'll take a look at the intense training T cells go through in the thymus boot camp. Then we'll see how a critical two-signal handshake launches the final attack. And, of course, we'll wrap it all up with some key clinical tie-ins and a rapid-fire board review to make sure it all sticks. Okay, so imagine there's a security breach at some high-tech facility. The very first alarm, that's going to trigger an immediate response from the general security guards on site, right? At the same time, a specialized team is being put together to handle the very specific threat. Your immune system? It works in almost the exact same way, with two totally distinct but complementary strategies. So which is it? Do you need the first responders, or do you need the elite snipers? The on-site guards, they're your first responders. They're fast, they're everywhere, but they're generalists, you know? They can handle a common problem, but they're not really equipped for some kind of sophisticated infiltration. The elite sniper team, on the other hand, well, they're highly trained, super specialized. It takes them a while to get there, but once they do, they are incredibly precise and effective. And that right there is the fundamental difference between your innate and your adaptive immune systems.
This table really lays it all out perfectly. Let's focus on those key differences. Your innate immunity, that's the rapid response team. We're talking neutrophils, macrophages. They are on the scene in minutes to hours. And their recognition is nonspecific. They use these things called toll-like receptors to spot very general, conserved patterns on pathogens, which we call PAMPs. So, what's the high-yield takeaway? It's fast, it's nonspecific, and it has absolutely no memory. Now, the adaptive system with its T and B cells, that's your elite sniper team. It is slower to get going, for sure, but it is surgically precise. And that's all thanks to something called VDJ recombination, which lets it create unique receptors for almost any antigen you can imagine. And, most importantly, it develops memory. This is the whole basis for how vaccines work and why you get lifelong immunity after some infections. Okay, so the innate cells have spotted a threat, but how do they tell the elite adaptive forces what's going on? I mean, they can't just send a text, right? They need a system to display exactly what the enemy looks like. And this is where the major histocompatibility complex, or MHC, comes in. Just think of them as cellular billboards advertising that there's an intruder and what kind it is. You know, at a quick glance, these two molecules might look kind of similar, but their structures really reveal their very different jobs. Over on the left, we've got MHC class 1. It has one long heavy chain and a smaller short chain. And on the right, MHC class 2 is made of two chains that are about the same length. Now, these aren't just random quirks. This design difference is directly tied to the very different functions these molecules have. Let's break that down. So here's the absolute core distinction. MHC class 1 is found on all nucleated cells, and that is a super important point. It presents endogenous antigens, meaning pieces of proteins made inside the cell, like little bits of a virus. It's basically the cell's internal distress signal, its way of screaming, "Help, I'm infected." And this particular billboard is shown specifically to CD8 plus cytotoxic T cells, the assassins of the immune system. MHC class 2, though, is only found on professional antigen-presenting cells, or APCs. Think macrophages and dendritic cells. These cells go around eating external threats. They chew them up, process them, and then display the pieces on MHC2. They're basically scouts reporting back from the field saying, "Hey, look what I found out there." And this billboard is shown to CD4 plus helper T cells, the generals who coordinate the entire adaptive response. And here it is, a high-yield memory hook that will absolutely save you points on exam day. The rule of 8. It's so simple, it's brilliant. Check it out. MHC class 1 binds to CD8 plus T cells. What's 1 times 8? It's 8. And MHC class 2 binds to CD4 plus T cells. What's 2 times 4? It's 8. It's just a simple multiplication trick to lock in these critical interactions forever. Seriously, don't miss these points. Okay, so we've been talking about these super specialized CD4 and CD8 T cells, but where do these elite soldiers even come from? they're not just born ready for a fight. They have to graduate from this incredibly intense and selective training program, a true boot camp that happens in a small organ right behind your sternum, the thymus. This image gives us the T cells' whole career path, right from being a new recruit to a seasoned warrior. They start out as precursors in the bone marrow, but at this point, they're just naive recruits. They then travel to the thymus to mature and get educated. And this is where they commit to becoming either a CD4+ helper or a CD8+ killer. But it is a brutal program, and trust me, not everyone makes it through.
So to graduate, every single T cell has to pass 2 life-or-death exams. First up, in the thymic cortex, is positive selection. The question is pretty simple. Can you even see our own billboards? Can you recognize self-MHC? If the cell can't, well, it's useless, and it gets eliminated. But if it passes, it moves on to the medulla for the second, even more dangerous test, negative selection. Here, the question changes. Do you react too strongly to our own body's proteins on those billboards? If the answer is yes, that cell is basically a potential traitor, an autoimmune cell. It's immediately forced to undergo apoptosis. Only the T cells that are both useful and safe get to graduate and go out into circulation.
Now you might be thinking, how does the thymus, this little organ in the chest, know what the pancreas or the thyroid looks like? Well, that's the genius of a protein called AIRE, the autoimmune regulator. For the boards, AIRE is a critical buzzword. It basically acts like a guest lecturer, forcing the cells in the thymus to express tissue-specific proteins from all over the body. This makes sure that any T cells that would attack those distant organs are found and eliminated. Now, if AIRE is defective, these self-reactive cells escape, and that leads to this devastating multi-organ autoimmune disease called Bأbsط. Okay, so our T cells have graduated. They're smart, they're safe, and now they're just circulating in the lymph nodes, waiting for the call to action. But how do we make sure they only attack when they're really supposed to? Well, to prevent a huge friendly fire incident, activating a naive T cell requires a very specific 2-part secret handshake. And here is that secret handshake. First, the APC presents the specific antigen on its MHC molecule. The T cell's receptor locks onto it. That's signal 1. Think of it as the initial hello. But that is not enough to launch an attack. For full activation, we need signal 2, the friend or foe password. This is a co-stimulatory signal, where the B7 protein on the APC binds to the CD28 protein on the T cell. Now here's the critical safety mechanism. If a T cell gets signal 1 without signal 2, it doesn't just fail to activate. It becomes anergic, which means it's permanently shut down. This is what prevents T cells from accidentally reacting to self-antigens. So, once an elite CD4+ helper T cell is fully activated, it can now authorize B cells, or little antibody factories, to join the fight. And guess what? This also requires a specific 2-signal handshake. First, the B cell presents the antigen it found to the already activated helper T cell. That's signal 1. Then, the T cell provides signal 2. It does this by using its CD40 ligand to bind to the CD40 receptor on the B cell. This CD40-CD40L interaction, that's the official green light for the B cell to start multiplying and, crucially, to start class switching, pumping out more effective antibody types like IgG and IgA. So we've traced this whole pathway of an immune response from the first detection all the way to the coordinated attack. But how does all this foundational science actually play out in the clinic? Let's connect this knowledge to the high-yield clinical associations and board-style questions you are pretty much guaranteed to see on your exam. Remember those MHC molecules? Well, in humans, we call them HLA, and certain versions of them are really strongly linked to autoimmune diseases. For your exam, it is absolutely critical to know these associations. HLA-B27 is famously linked to the seronegative spondyloarthropathies. Just use the mnemonic pair. HLA-DQ2 and DQ8 are classic for celiac disease. And HLA-DR3 and DR4, you see those pop up in a bunch of autoimmune conditions, especially type 1 diabetes and rheumatoid arthritis. Knowing these associations and the mnemonics will make these questions second nature for you. All right, let's put that knowledge to the test. Rapid-fire review time. Question 1. A virus infects a liver hepatocyte. What's the mechanism for clearing it? The answer is A. A viral infection is an endogenous threat inside a nucleated cell, so that hepatocyte is going to present viral antigens on MHC class 1 to a CD8+ killer T cell for destruction. Okay, question 2. A defect in the AIRE gene. What does that mess up? The answer is A. AIRE is absolutely essential for expressing self-antigens in the thymus, so a defect directly screws up negative selection. This lets self-reactive T cells escape and cause autoimmunity. Last one, question 3. A new drug blocks the interaction between B7 and CD28. What's the consequence? The answer is A. That B7-CD28 interaction is the critical signal 2 we talked about, the one needed for naive T-cell activation. Block it, and you get anergy, a failure to activate those T-cells. And that brings us to one final thought to chew on. We've seen how the immune system has this incredible challenge of distinguishing self from non-self. So how do cancer cells, which are basically mutated self cells, manage to exploit these very same checkpoints to hide from and evade destruction? Understanding that question, that's the key to the entire field of modern cancer immunotherapy.